Okay, thank you, Fabian. Hello and welcome, everybody. Today is my great pleasure and honor to introduce you to Mr. Klaus Lunau. Uh, Klaus is a very rare, passionate, and authentic naturalist. He has studied biology and done his PhD at University of Freiburg, and afterwards he worked as a professor at the University of Regensburg. And from 1997 to a couple of years ago, he worked in the University of Düsseldorf. And now Klaus is retired, but he's still very active. Klaus has published many papers in several research areas, including insect cognition, flower color, and of course, pollination. And Klaus is also very interested in pollen flowers. And I think that among all of them, melastomes are his preferred ones. So, Klaus, thank you for accept our invitation, and please talk, do your thing.
Yeah, hello, Melas enthusiasts. I'm probably the first one who talks in this seminar who is not an expert in Melastomataceae, and I'm even not a botanist. So you're gonna discuss today with a zoologist, and maybe it's sometimes a bit strange to you. So let me see how I can change to the next figure here. So what I want to share with you, my agenda is that what I read in textbook before I knew Melastomataceae was very different from that what I saw when I came to Brazil and visited Vinny about Melastomataceae. So this discrepancy about textbook knowledge and personal experience with flowers of the Melastomataceae I will share to you. And my agenda includes the absence of color pattern in the flowers of Melastomataceae, their radial symmetry, the fact that they are pollinated by buzzing bees, and the fact that they have unique stamens. First, I want to thank a couple of persons who helped me with my research on Melastomataceae. This is Andre in the University of Diamantina, and my doctoral student Sabine Konsmann and her field assistant. But first of all, I have to thank Vinny, who first introduced Melastomataceae to me, and almost everything I learned about Melastomataceae is from him. So it's a bit biased, the talk. So thank you, Vinny, in advance very much. So let me start with this largest survey of ultraviolet reflection pattern by Bohr and Bartlett, which included more than 6,000 angiosperm species, and also 43 Melastomataceae flowers were included. And they found that Melastomataceae have almost no color pattern. So among the 43 species, there were only 4 which have a color pattern in the ultraviolet range of wavelengths. Here are 2 of these examples. But for the rehabilitation of Barbara Bohr and Wilhelm Bartlett, I want to say that they at least acknowledge that they have frequently yellow display answers. So it seems to depend on the definition of color pattern. If we consider that Melastomataceae flowers have color pattern or not. So if we define color pattern as any visible color pattern to a pollinator, then Melastomataceae flowers are outstanding for its display of floral color patterns. But why do we know so little about color pattern in Melastomataceae? Let's look behind the curtain. So here you see fir and they are experiencing the flowers of Melastomataceae, in this case Tibucina pulchra, with a spectrophotometer, and you can see here the spectral reflection curves they get. But then you can also only measure the petals, but not the anthers and the stamens, because they are too small and because they are too rough, and they are not accessible to the spectrophotometer. So you have to choose a different method, and this is UV photography. And here again, Winnie experiences it, and I think it was Winnie who published the first false color photos of flowers at all, with these photos you see here about the floral color change in Tibucina pulchra. So it is largely dependent that the normal method, the spectrophotometer, is not accessible to Melastomataceae, and I encourage every researcher who wants to study color pattern in Melastomataceae to use alternative methods such as UV photography. I will come to this later in more detail. So first you have to learn something about these little yellow tiny dots, which you also find in the color pattern if we define this like we did in Melastomataceae flowers. And this was a large extent my work, that I found that the yellow color of pollen and anthers is caused by flavonoid protective pigments, and many flowers which have their pollen not visible, so the pollen is hidden in the anthers or the pollen is hidden in the corolla, they display pollen mimics or anther mimics. So these can be anther patches, lower lips, filamental hairs, fake stamens, flowers in combination with bracts, or disc florets in combination with ray florets. And this does not only hold for blue flowers, in which yellow is a very nice color contrast. This also holds, of course, for white flowers, and this also holds, of course, for yellow flowers. But here we have to consider the second aspect of these yellow central spots, which is the fact that they are always UV absorbing in the center, as you see here. And this probably also holds for many Melastomataceae, which have yellowish, whitish parts, at least in their stamens, as you can see here in these flowers. These yellow spots, they are very important to direct pollinators to the flower. This is a worldwide language for bees, which is understood in every continent. So it's a real bee Esperanto. I have checked this worldwide genus Utricularia, and here you can see in every continent you have these yellow spots addressing signals to bees. But what does it mean? So we have different kinds of stamen mimics. So real floral guides, disc florets, stigmas, lower lips, staminodes, and filamental hairs. And you see that up to 1/3 of the flowers in different floras display at least one of these types of pollen and stamen mimics. This is a bit lower in the neotropics because there's a large amount of flowers which are pollinated by bats and by hummingbirds, which are not interested in pollen, and so they are also not interested in pollen mimics. And almost everywhere, bees respond to these signals spontaneously. So if you test naive bees or experienced bees, you find similar results. They are approaching, when they approach the flowers, they will respond to the edge, but when they see these little tiny yellow dots, they will make contact with the antennal tips before landing. And this also holds for Melipona quadrifasciata, a stingless bee in Brazil, and also for solitary bees. And this aspect of flower coloration is very important for the advertising of a landing platform for the approaching pollinator. So in nototropic flowers, these stamen mimicking, pollen mimicking structures, they are mostly on the ventral part of the flowers. And in sternotropic flowers, these pollen mimicking structures, they are mostly on the upper part of the flowers. Those stamens and stamen mimics have a distinct relationship here. The stamen mimics are more conspicuous than the real stamens, and they direct the flower visitors to the nectar or to the landing platform. Otherwise, they would directly approach the stamen and probably be less effective as pollinators. So let's see here in a little video how this works. And here you can see a honeybee, which is experienced. And in this case, we wanted to check how tiny can these yellow spots be. And in this case, it's less than 1 millimeter. And the bee approaches these yellow spots with the antennae. And here you see its experience. Then it crawls up and extends the proboscis, to which the bee knows already that there will be some sugar water reward. But what we don't know is whether the same happens with Melastomataceae flowers. And this still has to be shown, whether Melastomataceae flowers have these yellow, probably always UV absorbing, conspicuous, contrasting against the corolla, attractive yellow areas, which direct the flower visitors towards it. And other parts of the anthers, they are inconspicuous, camouflage, and they are distracting the flower visitors from these parts. And probably this behavior, if it will be shown in the future, helps to increase the pollination success of these flowers. When I visited together with Winnie the last pollination congress in Brazil, in Catalão, we took a field trip, and then I found this Cambacidacea, a flower, and Melastomataceae. And this has a real strange color pattern. So it's very different from what I told you. And this reminds me that there are probably other functions of color. But in particular, it was very astonished that the petals have an asymmetrical color pattern. Each petal has an asymmetrical red spot at its tip. And my hypothesis is that it protects the pollen in the flower buds, because only these red tips, they appear above the calyx. And one prediction from this hypothesis that when you have small red tips, then you should have a large calyx, and when you have large red tips, then you should have a small calyx. This you can check even with photo material from the internet. So this is probably not an attractive device, but a protective device, maybe to shield the pollen against ultraviolet radiation, or to shield against herbivores. Again, Winnie was also part of a study which we did together with Gabriela de Camargo, and was published in New Phytologist. Here was a cover photo with some Melastomataceae. And in this graphic, you see from this study the reflection of the Melastomataceae, which were covered by this study. And I have also included, Gabi sent it to me for this talk, a color photo, a UV photo, and a false color photo of this Campesidaceae flower. And here you see that the red tips are not particularly conspicuous for bees, because bees are red blind, so these are almost black for bees. So there's no argument that this has a special attractive function for bees. What you can see here, that Melastomataceae flowers, they are not different in color from other flowers. We have only rarely yellow ones like this, but we have many white flowers or pinkish flowers, which have here either UV reflective or UV absorbing spectral reflection curves as shown here. So with a Hitchhiker's Guide to the Melas colors, I will remind you on the function of flower colors and flower color patterns. So as I mentioned already, attraction towards the landing spot, this is one of the main functions. Deterrence also. This was a part of the study with Gabi, that many red flowers deter bees because they want to be selectively attractive for hummingbirds, and also protective function against ultraviolet, for example. At the long range, the detectability is most important, so the flower has to stand out against the background. For the innate preference, there are distinct color hues and the color saturation. These are parameters which are known that bees respond to. Maybe also bees that visit Melastomataceae flowers. When it comes to learning, maybe all perceivable color parameters have to be considered, because bees are very clever learners, and they can learn almost every color parameter. As I mentioned already, but I think it's very important, if a flower has no color pattern, the bee will respond to the edge of the flower. So it will land here. And then it's not easy to find access to the nectar or to the pollen, or to be placed on the flowers in a proper way to pollinate the flowers. So the flowers, they have to guide the bees, the guidance towards the center, towards the reward. And this is what this color pattern, or the bee-colored stamens of Melastomataceae flowers do probably. And this is constrained by the spectral sensitivity. I mentioned already, bees are red blind, and these red petal tips are not particularly interesting for bees. And later I will tell you that bees are colorblind when they see flowers from a distance. Then only the green contrast matters. and they have some hardwired behavior. So they prefer to learn distinct color hues, but they spontaneously prefer higher color saturation. At least bumblebees and honeybees do so. So these are the important color parameters. So the distinct color hues, and for one color hue, here red, I have shown the different values of color saturation, here very low, here high, and here different values for brightness. Brightness is mostly not considered in research, but for the detectability, brightness seems to be very important. At least in nocturnal insects, they probably respond to the brightness of flowers. Many of flowers which are visited by nocturnal pollinators are white, as you know already. So what do we see and what does the bee see? So this is a flower of a forget-me-not, and we see here they have a color change of the stamen mimic from white from yellow to white. So these are the rotting, these are the non-rewarding flowers, and this is what a bee will see. And how do we manage to know what a bee can see? So here, this is a little Andrena vaga here on my nose. And to explain what the bee sees, we take a color photo and a UV photo. When we split the color photo into the blue, the green, and the red component, and we take a UV photo. And here on the UV photo, you see already that not only the central part of the flower, but also the petals have different UV reflectance. And then we reassemble the flower, and UV appears as blue, blue as green, green as red, and red is discarded. And this is the false color photo we get then. And here you can see the rewarding flowers are very outstanding in color contrast for a bee, and even the petals are different in colors for a bee, and we cannot see this difference with the human eye. So from a distance, a bee is colorblind. So only when a flower appears in a larger viewing angle, the bee will see colors, and they have a very low spatial acuity, so they do not see millions of pixels. They only see several thousands of pixels each ommatidium, and the honeybee has about 6,000 ommatidia, contributes 1 pixel. And maybe this is how these bunch or this inflorescence of the forget-me-not is seen from a distance. And this is very important for the flowers, so they have to provide green contrast, otherwise they are not detected from a distance. And only if the bee comes closer, then the bee brain also processes the color. Probably this is a consequence of the tiny brain. There's only very small brain. They cannot process color information for each ommatidium 10 times per second or so. So they rely on green contrast, and only when several adjacent ommatidia have the same information of green contrast, they process the color information. And then these tiny targets in the center become very important for the landing. So the next issue I want to talk about is symmetry. Many textbooks mention that Melastomataceae have symmetrical flowers, and I also believe that is so. When I prepared this talk, I found some interesting aspects of flower symmetry in Melastomataceae. I think they are worth sharing with you. So the first aspect, which is very strange, is when you see these radial symmetrical flowers, uh, they look a bit strange. So as if it should not be displayed in this horizontal way. Maybe it's better to display these flowers in a vertical position. And here you see you have both cases in one inflorescence. So this top flower is placed or presented horizontally, and these other flowers are presented vertically. And this is very unusual for flowers, that the flowers in the same inflorescence are displayed in different manners, so vertically versus horizontally. Of course, you have Melastomataceae, which are truly radially symmetrical. Here's one example, and others which are truly monosymmetrical, as shown here. But my experience about flower symmetry is from the Alps, and you can be sure Vinny also accompanied me there in the Swiss Alps and shared with me this experience. So this is also biased by Winnie's opinion, I think. And my favorite flower in the Alps is the starry saxifrage, Saxifraga stellaris. And there you have the same. You have one top flower, which is radially symmetrical flowers, and the flowers which stand to the side, so this is the top view here of the inflorescence, they are monosymmetrical. You can see here they have these large yellow spots, and here these other petals, they have tiny yellow spots. And that's why I became interested in flower symmetry and differences in flower symmetry in the same inflorescence. This is very strange. It has never been mentioned in the literature that this happens. So we have here another example shown at the close-up. So more or less radially symmetrical flower, and here flower which has only little yellow spots. So it appears more or less monosymmetrical. In the textbooks, these flowers are defined as radially symmetrical, which is obviously not true. One aspect is that when you check the terminal flowers and the flowers which are presented horizontally, they are mostly radially symmetrical in saxifrages, in Saxifraga stellaris and other saxifrages. And the flowers which are displayed in a diagonal or in a vertical manner, they are mostly monosymmetrical. And the features which are defining this monosymmetry are the color pattern, I mentioned already, the stamen movement. So the first stamen that moves, they move, all the 10 stamens move one after the other. The first stamen that moves is almost only that from the basis, which is facing downwards, and these stamens which are facing upwards, they move last. And the interpetal angle can also be a difference. So here the interpetal angle is 72 degrees, so which makes up a monotonous radial symmetry. And here you see the interpetal angle is increased and is defining or is contributing to monosymmetry in this saxifrage. So what has this to do with melastome? So here are other examples. So in the Saxifragaceae, we have flowers which are almost good examples for radial symmetry. We have some rare examples of Saxifragaceae which have this monosymmetry in all flowers. And in these examples, we check that we have this variability within the same inflorescence, so in the same individual, where we have monosymmetrical and radially symmetrical flowers. And in most cases, the color pattern and the angle between the um, petals is involved. So here in this Saxifraga rotundifolia, these petals have intense color patterns and these petals have only pale color patterns. And in Tibucina, in melastomes, you find a similar example. So here, this flower is presented vertically, and in the same inflorescence, you find flowers which are presented horizontally. So this is a shared phenomenon with the Saxifragaceae. Is here the evolution of flower symmetry on the way? What seems most likely to me, as when I checked Melastomataceae flowers for this purpose, what Melastomataceae are unable to do, because they are obviously constrained, is they cannot vary the angle between the petals. So the petals seem to be almost organized in a radially symmetrical manner. But they also, just as for the color pattern, they use also the stamens to make up a radially symmetrical flower or a truly monosymmetrical flower. So I think this is a constraint other than the Saxifragaceae. They have to do everything what makes up the visual appearance with the stamens. So they are outstanding for this. And are the stamens so extraordinary? And I asked this question in the context of the fact, how do the flowers sense where is upward, where is downward, how do they feel how they are oriented? So you can think that the flowers, sorry for this typos, the flowers can sense this information from the incident light, but this is obviously not true. At least we have some experimental evidence from Saxifragaceae that's not true. They can sense this information from gravity. In Saxifragaceae, we found that. So if you place the flower pots on a rotating vertical wheel, the flowers that develop are more radially symmetric. So when they have no constant gravity information, they will develop more radially symmetrical. And they also use information from the position within the inflorescence. We have some evidence in Saxifragaceae too. But here in the Melastomataceae, the shape of the anthers and of the stamens is very interesting. And I think that there are convergent examples like this. Cassia has also these curved anthers and stamens, and also this Duanthera, which I saw in China, in Kunming, from the Asparagaceae family, has also the appendages and the anthers very similar to that in Melastomataceae. And therefore, I think it could be that the center of mass is defined by these anthers, and they passively use this kind of information from the center of mass that the stamens or the anthers are hanging downwards out of the flower, but has never been tested. When I prepared this talk, this literature came to my eyes. Here's again Campesidaceae, and here you see young flowers. The stamens are still folded in the corolla. Then the flowers are, when they open, they appear more or less radially symmetrical, and only later they rearrange their stamens and then are monosymmetrical. So this is from Malis together with a student, published in in Portuguese, the rearrangement of stamens, but the mechanism how this is done has never been tested, to my knowledge. So it's in the textbook is always considered that Melastomataceae are pollinated by buzzing bees, but I got funded from the Deutsche Forschungsgemeinschaft for a project about the fine-tuning of the allocation of bees. So why do some flowers exclude some bees and only avoid distinct bees? And this seems to be in particular the case in some Melastomataceae flowers. They are not pollinated by all buzzing bees and not at all by all bees. And we tested this. This is work by Sabine Konsmann together with André Rech in Diamantina, Minas Gerais. And the flowers of Rhynchianthera grandiflora, which you see here, they have heteranthary. So they have 4 short stamens and 1 large stamen here. And yeah, let's see what we found out. So first, we checked the color pattern. And in this case, the stamens are more or less camouflaged. At least they are not outstanding yellow and UV absorbing as in other cases of stamen mimicking devices, but the yellow UV absorbing part is here in the center of the flowers. So maybe the bees are directed to fly deep into the flower before sitting down. So here from the side, it is obvious that the anthers are more camouflaged than conspicuous. And here from the color pattern in the false color photo, you can see similar, that this yellow center is mostly outstanding and conspicuous, directing the bees to the center part. And what we found here is that the bees are, the pollination success of the bees is depending on bee size. So we have bee robbers, we have bee robbers which are not pollinating. They break up the anthers, they are destroying the anthers, and they collect pollen without touching the stigmas. These are some Trigona bees and some other little bees. And then we have middle-sized bees like these Aurochloropsis and Melipona bees, and they buzz single anthers, so not all anthers together, only single anthers, mostly also without touching the stigma, though these are not pollinators. And the flower would benefit to exclude these bees from visiting the flowers. And these are the pollinators. So these are large bombus beans and Chilocoba bees, which grasp all the anthers and then buzz the flowers. Yeah, here we see the foraging events, and you see the small bees. They deal with the short anthers and with the long anthers. So they visit all these anthers, and the large bees and the middle-sized bees, they grasp the short anthers and they ignore the long anthers, and this is what obviously benefits the flower. So when we investigated this single visit pollen deposition, so how many pollen grains are deposited on the stigma by a single visit, then you find that the small bees, they do not deposit much pollen on the stigma, but the large bees, they deposit about 50 pollen grains per visit on the stigma. So This is what you find in a single visit, about 50 pollen grains or maximum of 50 pollen grains deposited on a stigma. When you have uncovered flowers and count how many pollen grains are delivered to the stigma in the whole flowering period, so this is about 150. And even if you glue the anthers so that self-pollination is excluded, you get a similar success. So they do, the bees do not contribute to self-pollination. This is almost always cross-pollination. And when you cover the anthers, then you have no autonomous self-pollination. So then it's astonishing, these very strange shape. So we have here the filament and here the anther. The anther has a rostrum and a tip. And in an experiment, we cut the tip, we cut the rostrum, and so we got 3 variations of these anther here. So these are the rewarding anthers, the feeding anther, and this is the pollination anthers, and this is the anther which we manipulated in our experiment. So here you can see what happens when you bust these flowers with an electric toothbrush. And here you can see how the pollen is sent out very directed from the tip, which like a little spoon. And once again, I like this video. Oops. So when you have here the long anther, then you see it's very directive, little angle where the pollen is spread out. When you cut the tip, then the release of pollen is very diffuse. And when you cut the rostrum, then the pollen only falls down. And when you imagine how the bee sits on the flower with these long anther on the back, then in this situation, the pollen is deposited on the bee's back. In this situation, the pollen is mostly deposited on the underside of the wings, and here pollen is almost not deposited on the bee. So this rostrum and the tip is a device, or these are devices to deposit the pollen targetedly on the dorsum of the bee. So we know that this is a so-called safe site, though the bees are not able to groom on this area so often. And maybe the pollen remains longer on the bee and thus can contribute better to pollination if it's is distributed here on this so-called safe site. So what we know already, that stamens can have different features, different functions. So they contribute to the color pattern. They can be feeding stamens. They can be camouflaged as pollination stamens. They can provide grip for pollinators. I think Winnie did some experiments about this. They help to dosage the pollen release, and maybe they can also impede the rubbery due to these foldings here, which makes it a bit harder for a trigona bee to cut open these foldings to get access to the pollen. So this was the time, 2012, when I visited Winnie in Pisciguama. Here we are standing in the Atlantic Ocean. We were protected against snake. You can see here our snake protectors. And at that time, we felt so good. We were protected against everything. And so we developed some crazy ideas. And I will talk in the last part of this seminar about the crazy ideas of the Winnie, which are not so crazy if you think about it. So this refers to my title photo. The Tibucina pulchra flowers, they are flowering in forest gaps. And in this case, the trees are very close together, but they can stand kilometers apart. So this is a distance like from Earth to Moon for a trigona bee. This can only be managed by a Xylocopa bee, and therefore it's important for these flowers to address their signals to these large bees. But maybe there's even more in it. So just to remind you again, trigona bees, they cut open the anther. They can get out the pollen from the anther with their proboscis. And without taking flight, a bumblebee and a honeybee cannot do this. Without taking flight, the trigona bees, they deposit the pollen into their cubicula. So they sit and remain on the flowers until the cubicula are filled. And when Winnie and the others were doing their field work, I was sitting and watching the same flower the entire morning. And I think it was not marked, but due to the regular appearance, it was the very same individual which visited this flower the entire morning. Now, these pieces have already been kept. It took hours before every stamen was opened and the bees fly back to the nest, come again, cut open the next stamen. So this was a job lasting more or less one day. So the entire flowering period. And the flower probably is not pollinated at this time. So this is a one-day job of a trigona bee. Yeah, for this opportunity, we took a UV photo. This is a bad quality because it was very shaky, very windy at this day, but I managed to make a UV false color photo. And you can see that the Tibucina flowers, they are standing out here, and you can almost not discriminate between the purple or pink flowers and the white flowers. So in the false color photo, they look very similar, and they are very conspicuous for bees over a large distance, and that's why we entitled this publication "Trees as Huge Flowers and Flowers as Oversized Floral Guides." So they operate over other distances as normal flowers. So when you remember, the small bee is visiting the same flower all the day. So what it can do is probably for pollination is probably nothing. And if it does something, it's only selfing. A medium-sized bee, which can visit some flowers, and maybe it changes the tree because it finds another tree when it empties the flowers of one tree. So it can contribute a little to outcrossing. And a large bee will probably visit one tree and another and another and another. So it carries the pollen grains of trees which may be positioned very far apart from each other. And this offers the opportunity for female choice. And this was the idea that Winnie and I, we discussed, and I want to to deepen this this aspect a bit. So when we think that the bees have an area where they collect pollen, so in this case, the ventral area of the bee, they have often specialized hairs which are branched. They hold the pollen very good. They are filled first at the bottom and then up the top. And they are groomed from time to time. Whereas on the non-collection side, here on the back, the bees have simple hairs just to warm the bees, but these hairs are not specialized for pollen collection, and probably they are also not groomed so often. So here the pollen, there are less pollen, but the pollen are not so densely packed here. And the pollen are less stratified than here in the pollen collection site. So when you now think that the stigma scratches over the pollen collection side and over the non-pollen collection side, then you can imagine that the stigma may get here only pollen from the last visit, so only mostly probably self-pollen or cross-pollen. And here the stigma gets pollen from a large array of last visits, maybe from different plants. And this offers the opportunity for female choice. So pollen delivery on the stigma from different mating partners, and then the female aspect of the plant has to decide which pollen tube growth it will block, or which pollen tube growth it will help that it can grow faster. And maybe this offers the opportunity to female choice. Yeah, that's it already. I thank you for listening and for your attention.
Thank you, Klaus. Thank you for this amazing talking, and also for these so nice pictures. And now, guys, I think Klaus will be very happy to answer your questions. You can unmute yourself or either put some questions on the chat. Klaus, there is a discussion here in the chat about the color pictures of the bee color picture.
Yeah.
Maybe it's nice if you can explain it a little bit more.
Yeah, I have already prepared one slide for this. Here I have this Asparagaceae, which looks very similar to Melastomataceae flowers. And what I've done here is I have a color photo and a color checker, and here I have a UV photo, and here I have then artificially introduced a UV absorbing and UV reflecting part. So of each color of the color checker is divided into a UV absorbing and UV reflecting part. And then you take both photos with a machine which is sensitive to ultraviolet light. So you have to block UV and infrared here. And here you have to selectively take a photo with UV light. Then you split. This is done easily by some programs like ImageJ. You split these photos in the blue, green, and red component. Or the UV photo has also to be split in the red, blue, green, and red component. And then it depends on the machine. So in my case, this was a Lumix camera in which the blue sensor is most sensitive to ultraviolet. In other fabricates, it can be the green sensor or the red sensor, which is more sensitive to ultraviolet. So then I discarded here from the UV photo the red and green part, and from the UV photo I discarded the red part because we do not need the red information. Bees are blind to red light. And then I rearrange the photo, I merge it as a false color photo, and ultraviolet will be blue, blue will be green, and green will be red, and then you get this kind of false color photo, which in false colors tells you what the bees might see. If anybody wants information about that, this is published by me and Christian Verhoeven, so I can send you the publication if you want.
Klaus, there is a question on the chat by Norbert Holstein, and he's asking, are the pollen safe sites pretty much similar across bee species, or can some taxa harvest as the wholly dorsal abdomen?
I think this is not investigated systematically. We did it for bumblebees, Bombocercis, and for honeybees. And what we did is we placed bumblebees and honeybees in a Drosophila tube and together with some pollen of wind collected, of wind pollinated flowers, so pinus pollen, for example. And when they start flying off, the bees are dusted completely with this pollen. And then we let the bees groom for half an hour or so, and then we check the pollen grains which remained on which part of the bee body. And then we find that because of the size of the legs, in the dorsum, there's in the middle one small area on the thorax, on the head, and on the abdomen, which is not groomed very effectively. And the same is true for the ventral part. So between the legs and also in the tail, between thorax and abdomen, the bees cannot groom very well. But of course, it's hard for a stigma to get into this area of the tail of a bee's body to get access to this pollen. What we also check is whether the bees sense where the pollen is deposited. So we targetedly deposited the pollen either on the head, on the thorax, and on the abdomen. And what we found is the bees don't care. They have a cleaning procedure or a grooming procedure. They begin with the head, then thorax, then abdomen, then legs. It's always the same. So only when you cover the eyes, they immediately start to clean the eyes, but this is the only exception. So this is a more stereotypic behavior.
Yeah, Norbert had a comment here. He said that it would be interesting to see how, for example, small bees cope with that situation. Maybe their legs are longer than and reach more.
Yeah, it has not yet been studied whether there are bees which have different behavior. So what I learned, for example, about euglossin bees, that they smear the nectar together with the pollen all around their body and then groom it again, maybe to desiccate the nectar. But this has nothing to do with pollen collection, I think, this behavior. It's more about nectar collection.
Okay, there are a couple of questions, and please feel free to ask directly to Klaus, but I can read as well. Fabian, do you want to say something?
Yeah, Klaus, quickly, a lot of the paradigm and melastome flowers is that they have to be buzz, and you've shown that there are different things, but Vinny have shown and Agnes have shown and Ricardo has a couple papers on that too, that as we get a higher elevation, there's other mechanisms, and one of the things that we see a lot are that the flowers no longer have poricidal anthers per se. They become more broader, the anther pores, but you still see bees coming. Is that a completely different type of behavior they have to approach how they approach those flowers, or you have any ideas there? Because another interesting thing is that most of those flowers at high elevation that have broad pores are now polysymmetric. They are no longer monosymmetric like the ones at low elevation.
Very good question. I don't know a really good answers. What I know is that the existence of a poricidal anther is not a precondition for budding the flowers. So we have also here in Germany, we have some flowers which have not a poricidal anther, but which are buzzed. And when I did my doctoral thesis, so this is about 50 years ago, there was one bumblebee. I did my doctoral thesis, bumblebees in the lab. And there was one bumblebee, which was red 69, I will never forget, which came to my hair and it buzzed my hair. And at that time, I had more hairs and longer hairs, so like this. And this bee did so for several weeks. And from this time, I got the impression when the bee is hanging, then this is the stimulus. When it hangs on my hair, this is the stimulus to start buzzing. So maybe there is some mechanical input, and I don't know how these flowers are. Are they erect and upright, or are they hanging? Maybe in hanging flowers, like in Solanum docamar, then the bees start more easily buzzing than on flowers which are upright.
Klaus, there's a couple of questions here in the chat. The first one is from Peter Quackenbush.
Yeah.
And he asks, does red provide green contrast for bees? There are a few red bee-pollinated species of Medinilla in Melastomataceae.
Yeah, these color parameters, which are you can see here on the screen, these are color parameters which are defined by a single color. But color contrast is always defined by a second. So it may depend on the background. So if the background is very bright, then a red UV absorbing color can stand out like a green contrast from the background. And if the red is UV reflecting, which is also colorful for bees. So this was, I remember, I mentioned the study with Gabriela de Camargo. This is a major difference between red flowers which are pollinated by birds and red flowers which are pollinated by bees. The red flowers which are pollinated by birds are mostly UV absorbing. So for bees, they are dark, black, inconspicuous, not very attractive. And red bee pollinated flowers, they are mostly UV reflecting, though for a bee they appear bee ultraviolet. And the hummingbirds don't care whether the red flowers reflect or absorb ultraviolet. But for the bee, it's important. But as I said, for the green contrast, the background is important. Many studies which are done now, they assume a standardized background. And I think it's time now to work with a real background. So when you have a flower which is flowering on the sandy white beach, it has a very different background from a flower which is flowering in the deep rainforest where it's dark against the bark, maybe a cauliflower, a flower which is flowering against the dark bark of a tree. And it matters for the bee and for the green contrast.
There is another question from Agnes. Agnes, sure you don't want to ask?
No, I can ask. I have like a million, like so many questions. No, thanks for the great talk, Klaus. One thing that I wanted to mention in the picture that you showed, you called the species Clidemia rigida. I'm very sure it's pollinated by some nectar drinking animals, and definitely not by buzzing bees. I saw a lot of nectar in these flowers. Okay. In the field, and it's probably I think it's rodents coming, but let's wait and see. But I was wondering now, do you know anything about differences in color perception of different bee groups? Like bombus, Xylocopa, versus then non-buzzing bees like trigona bees. Is there anything known about this?
Yeah, I think we know the spectral sensitivity curves of some bee species, including the western honeybee, some bumblebee species, and also some stingless bee species. But they are not very different. So in many studies, the spectral sensitivity of bees is approximated by the spectral sensitivity of honeybees, and maybe this is not a big mistake. We know one exception. There is one bee which is neotropical too, and it is I forgot the name at the moment. It is an oligolectic bee which collects pollen from only one group of reddish pinkish flowers, and this bee has developed a red receptor, so it's tetrachromatic. And maybe this is in the context of the oligolectic. So the bees can, but most bees obviously do not.
But it's really interesting when you think about how specific scent compounds, for example, can be an attraction of specific species, and then color apparently is not as specific or not as species specific. This is really, yeah, interesting.
Yeah, so I think the most common mistake which is done by researchers who are not so firm with color preferences, they think that color is color hue, but color has more information than color hue. And what I found is that color saturation is very important for bees, and color contrast is very important for bees. And maybe only in the second place, the color hue, so whether a color is yellow or blue or green, is matters.
And related to that, I mean, I know the work that you did about the papillae on petals. Have you checked several melastome flowers? Because I mean, I've seen that the petals have very large papillae if they are bee pollinated, and then when species shift to vertebrate pollination, petal surfaces are just flat.
Yeah.
So I wondered whether you have differences also with different bee pollinators, probably.
Yeah, we have one paper which is accepted now by Journal of Pollination Ecology and will appear soon, in which we made casts of petals. So
cool.
to exclude the odor, we made casts, and we checked by microscopy that you cannot distinguish a cast from a real petal if you use this material what dentists use. So you can fabricate really good casts. And for this purpose, we used Dibucina urveiana, and we the upside of the petals has papillate epidermis cells, and the underside has flat epidermis cells. And then we experimentally check with different light conditions, so diffuse light conditions or spotlight against the direction of the bee or in the direction of the bee. And we found this is very important for the spontaneous choice. So when the bees can see the gloss, which is erased from the flat petals, then the bee avoids these flowers. It prefers the flowers which produce less gloss, which due to their papillate epidermis cells.
Wow, really cool. It's great to see that you're actually not retired.
No, this is a long-standing manuscript, so I think I submitted it before I was retired.
Okay, cool, thank you.
Klaus, I have a very general question for you.
Yeah.
So I'm just wondering why in Melastome some basic floral functions as attraction and symmetry is more related to the stamens than to the petals. So
yes.
Do you have any guess?
Yeah, my best guess is constrained. So maybe the flowers are unable to develop different angles between the petals. If this is the case, then they have to manage monosymmetrical flowers with other structures, and melastomes do so with stamens. Yeah, I think it's a constraint, maybe.
Okay, thank you. Do we have any more question? Or it's time, Fabian.
Well, I could be here for 5 days. So
yeah, me too.
But I think this is really great. Thank you, Klaus. Amazing. We'll be putting the recording soon on the YouTube channel for anybody who came in a little late. And or if you want to rewatch it, there are a couple of things that I want to look at it again. Thank you very much. And we'll be announcing the seminar for next month shortly. We have to finalize a couple of details, but we'll be announcing it shortly, the next couple seminars. So thank you so much. And again, if you want to volunteer to give a talk, contact Vinny, Agnes, Lucas, and Flavia or me, and we'll be happy to include you in this rotating disco party. Thank you very much.
Thank you all. Bye.
Thank you, Klaus. Bye.
Bye.
Bye.